Hello everyone. Today I'm going to talk about DeepPLIER, a deep learning approach to pathway level representation of gene expression data. My name is Yichen Zhang. I'm a PhD student from University of British Columbia, Department of Statistics. This is a joint work with collaborator Bernard, Maria, and my advisor Sarah Mostafavi. One important task in gene expression analysis is to learn latent variables from it. Starting from a high-dimensional gene expression matrix, we hope to get its lower-dimensional latent representation from some latent variable models. These latent variables are generally a mixture of information, including biological signals, like cell types, pathways, and environmental factors, like age, diet, and sex. Some can also be technical factors, like batch effects. One challenge in latent variable learning is to identify biological signals from this mixture of information, while reduce technical factors. In a recently proposed method called PLIER, the authors proposed to incorporate prior knowledge, such as pathways and gene sets, in learning latent variables. The key element of PLIER framework is aligning latent variables with known pathways and gene sets. By such design, biological signals, which usually have correspondence in prior knowledge, are preserved in latent variables, and technical noise, which usually vary study by study, are filtered out. However, one limitation of PLIER framework is that latent variables are constructed as a linear combination of genes, in other words, by a linear model. Because of that, nonlinear associations are not captured by such model. At the same time, it is well known that biological systems are complex, where different genes can interact with each other in a nonlinear fashion. And some genes can have interactions with environmental factors as well as latent variables through a complex network. The motivation of our work, DeepPLIER, is twofold. First, inspired by PLIER, we want to incorporate prior knowledge, such as pathways, in order to identify biological signals and to reduce technical noise. Secondly, we would like to capture nonlinear associations where the PLIER framework does not allow for. We propose to use an autoencoder structure for latent variable learning. The nonlinear associations are captured by having nonlinear activation functions at each layer. Different from PLIER, we incorporate the prior information in two parts. Firstly, we have those colorful nodes which denote pathways. These pathway nodes are only connected to the genes belonging to each specific pathway. On top of that, we have fully connected nodes, which are connected to all genes. These fully connected nodes allow the network to learn important latent variables when some pathways are misspecified or partially missing. In addition to that, we further impose positive weight constraint at each layer to ease model interpretation. The first experiment we did is to assess how well different models can predict latent variables from simulated gene expression. We compare our model DeepPLIER to PLIER and took PCA as a baseline model, which does not take advantage of the prior knowledge. From the first column, where complete pathway information is provided to both PLIER and DeepPLIER, we observe that the DeepPLIER produces more accurate latent variable prediction than PLIER does. More interestingly, from the second column, when only 30% of the pathway information is provided, DeepPLIER still outperforms PLIER, and some latent variable predictions remains high. This indicates that DeepPLIER can handle cases for misspecified or missing pathway. Secondly, we move on to the task of cell proportion estimation on a real bulk brain tissue expression dataset from ROSMAP study. ROSMAP study is a prospective longitudinal cohort of aging population where bulk brain tissue RNA-seq is available. Here we focus our attention on a subset of those individuals for which cell proportion estimates of 5 major cell types available in a 2019 paper by Patrick et al. We compare DeepPLIER to PLIER and the other 4 methods which are specifically designed for the task of cell deconvolution. Looking at the plot, we observe that among 5 major cell types in brain tissue, DeepPLIER gives the best cell type proportion estimation on 3 cell types, namely astrocytes, endothelial cells, and microglia. Overall, DeepPLIER latent variables can capture cell type signals from bulk tissue data better than PLIER and the other 4 methods in this experiment. The replication of latent variables across datasets is a good indication of whether the learned latent variables are capturing biological signals or just a technical noise. This is because the technical noise can vary study by study and are usually not replicable, while the biological signals can be correlated by sharing some common pathways. In this experiment, we assessed the replication of latent variables independently learned from 2 brain tissue expression datasets, namely ROSMAP and Common Mind. The DeepPLIER was compared to PLIER as well as 2 baseline settings where the pathways are either all misspecified or not used at all. From the plot, we can see that DeepPLIER produces more replicable latent variables than PLIER does. Comparing DeepPLIER to 2 baseline models, we can know that correct pathway information helps the model to extract biological signals. Here is an example of how DeepPLIER latent variables can reveal biological insights across datasets. Looking at one of the highly correlated latent variable pairs from ROSMAP and Common Mind, we found 3 common high-impact genes, which have been shown to be related to microglia activities. This is in agreement with recent findings about the role of microglia functions in Alzheimer's disease and schizophrenia. For example, proliferation and activation of microglia in the brain is a prominent feature of Alzheimer's disease. Microglia degeneration caused by inflammation is a potential mechanism of schizophrenia. In summary, we have shown that DeepPLIER can better handle pathway misspecification than PLIER does. DeepPLIER latent variables can capture cell type signal from bulk tissue expression. Latent variables extracted by DeepPLIER on real data are more replicable than PLIER. DeepPLIER latent variables can reveal biological insights across datasets.